Okay, so you should be able to see my screen now. I hope it's the right one with the presentation display. Wonderful. Yeah, so like Christophe already said, my name is Hanna Spitzer. I'm from the Computational Health Center in Helmholtz, Munich, and I work in Fabian Theis's group. So we've been developing with a large community, actually, these Python tools for single cell analysis. So you may have heard of Scanpy, and recently me and Giovanni Palla were kind of leading the development of SquidPy, which is this Scanpy extension for spatial omics analysis. But I'll be talking about SquidPy and spatial transcriptomics tomorrow. So today I'll just be talking about kind of, best practices in single cell omics data analysis. So this will be mainly focused on QC and on data analysis. And I hope it's not too basic for you guys. So please, if you have any questions during the talk, please feel free to interrupt me and ask at any time. I've just tried to collect kind of basic best practices that we are using ourselves in the lab, and that we see that the community has kind of converged on when using this single cell data. I want to mention that these slides, they're actually mostly compiled by Anna, who will also actually appear in this course, I think also tomorrow or maybe the day after on the cell-cell communication. So you can also look forward to this. These great slides, they're actually by her. So thanks for letting me use them, Anna. Okay. Oops. How do I switch? Sorry, I'm trying to find my cursor so I can actually switch the slides. Yes, wonderful. So this is a basic overview of what you essentially do with single cell RNA sequencing data. So first off, we start with a feature matrix. This can be, this can contain loads of different types of data. So traditionally, maybe in our lab, we mainly use RNA sequencing data, but it can also contain some spatial information, and it can actually be expanded to, for example, ataxic or site-seq data, or even more types of modalities. But ultimately, what you have is you have a matrix that is cells by features. This could be genes or proteins or whatever features you're looking at the moment. And from these matrices, we would like to ideally gain some information about the biology. And in order to do this, there's a couple of steps that one needs to take for processing this data. And the first step is the preprocessing and visualization. This is a lot of quality control. There's normalization of the data, and then of course some visualization, some, for example, UMAP plots that you can do. And after you have preprocessed your data, you can go on to identify, for example, different groups of cells that you may have in your data. So this is usually done with a manual annotation workflow, but more and more there have been novel tools that allow you to also do this in a more automated fashion by, for example, using large atlases and kind of transfer these annotations that you have in these atlases already to your to your individual samples that you're looking at. And after these are kind of the basic preprocessing steps. So the preprocessing and the cluster annotations. After this comes the actual interesting part, let's say, of the analysis, which would be you could look at one modality only and, for example, look at highly expressed genes or differentially expressed genes in certain conditions. Or if you, for example, have a developmental data set, you could look at trajectories, maybe using RNA velocity or other methods to kind of infer some trajectories, some directionality in your cells. Or there's another type of methods that tries to look across the modalities. This is quite a new field, so there is not a lot of established methods for this, but essentially you could use some machine learning or some variational autoencoders, for example, to learn a unified representation of your data, and then do some subsequent analysis and visualization of this kind of unified space from all of your modalities. For all of these tools, there is this quite useful website, single cell RNA tools, that you can have a look at to get an overview of the single cell RNA software ecosystem that there is. And you can see that the number of packages, this is both Python and R packages, has grown kind of exponentially, at least more than linearly in the last years. There is currently like 1,200 different tools, and there's many different categories, of course, for these tools. But the main categories would be visualization, clustering, dimensionality reduction, integration, and all of these basic things that you need to do to process your data. So you can check this out if you're interested in discovering maybe new tools, or if you want to add your tool that you have developed to the database for other researchers to allow this to see them. But essentially, this also means that this whole ecosystem, there is a lot of tools, and it might be very overwhelming to understand which tool is the one you should use for your particular question. And for the Python ecosystem, we have recently launched this scVerse, which is supposed to be like the analysis ecosystem that you use if you have single cell data and want to use Python. So there's tools like AnnData and Scanpy, of course, in there, but also MuData and Muon for multi-model data analysis. And then more advanced tools like SCVI tools for integration, SirPy for looking at immune data, and SquidPy for looking at spatial data. And we are looking to add more and more tools to this community to give all of us a way of organizing the packages a bit better. Today, I'll just be focusing on AnnData and Scanpy, because these provide essentially the most basic preprocessing tools that we can have. Okay, so like I said, mainly I'll be looking at preprocessing and visualization and clustering, and maybe more in detail what this entails. I've already introduced this a little bit, but essentially what you do with your data is you need to generate it, usually in the lab. I mean, not usually, this is how the data is generated. I'm a computational scientist, you may be able to tell. I'm not in the lab myself, so the data essentially starts when I get this count matrix from the actual experimental data. So this count matrix, then there is a quality control step that we need to take, and I'll be walking you through some examples and some guidances and best practices of how to do each of these steps. After the quality control, there's the normalization step, and after this we can, for example, reduce the dimensionalities if, for example, we only want to look at highly variable genes. This is a very common step that one takes with this type of data. And after this, we can do visualization with different methods. And then after this, like I said, after this preprocessing, we are looking at identifying different clusters of cells, at annotating them with biological meaningful names, because ultimately we want to be able to interpret whatever effects we're seeing. So we need to know what kind of cells are we looking at here. And then finally, after you've done this, like I already said, you can do stuff like differential expression, trajectory inference, or model, for example, the effects of different perturbations that may be occurring in your data, and figuring out which genes are responsible for what perturbation effect that you're seeing. And of course, you can, from this data, you can also look at cell-cell communication inference, which is also something that Anna will be looking at. I think it's tomorrow afternoon or on Thursday, and it will be showing you how to do this. Right. Okay. So the main data structure that we have is, or that we're using here, is called AnnData. This stands for annotated data frame. And yeah, essentially it's exactly this. So we start with this X matrix, which is our counts by genes, or more general counts by, sorry, not counts by genes, cells by genes, or cells by features matrix. And it contains as entries, if we stay, let's say, in the single cell RNA sequencing world, this would contain in every cell the number of counts for this particular gene that occurs in this particular cell. So this is the X. And then you can have additional information on top of this, on top of this X data frame, which can be the OPS, the observation matrix. So this would be cellular level information. For example, this can be which sample does the cell come from? It could be the cell type of this particular cell, or other information. Maybe, for example, you've calculated some cell cycle score. This could also be contained in the OPS matrix here. So these are either 1D or 2D data information. So there is, in addition to OPS, which is just a pandas data frame, there could be also OPS-M, which is then a matrix that is also has as common axis as the cells. But for example, if we're talking about spatial data, it could actually contain the X and Y coordinates of where your cells are coming from in the particular tissue. So this could be obtained, could be stored in OPS-M here. Yeah. In addition, we have the var matrix, which is the annotation of the features. So this could, for example, contain more information about the genes. So it could contain the actual gene name or the ensemble ID of the genes. It could contain information whether this gene is highly variable in your sample, or whether it's differentially expressed in a particular cluster. Whatever kind of information you want to store over the genes can be stored in var. In addition, there might also be some unstructured information. This is stored in ans. And this is essentially just a dictionary. So you can store whatever information that does not fit over the axis of variation of variables or of observations. You can just store in ans. So for example, what is usually stored by Scanpy in there is, for example, the colors that you want your clusters to show when you're looking at a UMAP. This is usually stored in ans, or any other metadata that any tool that you executed on your data, anything that is useful for reproducing your analysis is usually stored in the ans. So an example of how this AnnData would look like, you can see here. Essentially, if you have an AnnData, an annotated data frame, you can print it, and you can see in this particular data frame we have 1,000 cells and roughly 12,000 genes or variables here. And in the observations, we've stored things like the number of genes, so the number of genes expressed in every cell, the size factors for every cell, which is used for normalization, and we'll come to this later. And for example, the calculated cell cycle phase, and yeah, other things like this. In the var, we've stored the number of cells that are expressing every gene, things like whether this gene is highly variable, and so on. And actually, you will observe that in Ops M, which is also seen here on the screen, the matrices, some of the matrices start with an X. This is essentially a way of representing that this is a derivation of our original data X. So for example, you have XPCA. This is just a principal component. I would assume maybe it's like the first 50 principal components of your data matrix, or XUMAP is a UMAP representation of your data matrix. So you can easily use these information to then visualize and plot your data. In addition, the AnnData object can actually have different layers. So here it has one additional layer, which is called counts. This is essentially a way of storing different data matrices. So in this AnnData, in X, you will have the already normalized gene expression data. And in the counts layer, you will have the raw counts that you got from your sequencing protocol. So this is a way how you can store also raw data, because actually these counts are used by some of the processing methods, like for example, doublet detection. You actually need the raw counts and not the normalized data matrix. So this is useful to also know where this is stored. Right. So yeah, please again, if you have any questions, feel free to interrupt me. I don't see the chat here, so if you want to ask a question, just unmute yourself and yeah, tell me. Otherwise, I'll continue with the actual pre-processing steps that one can take and some best practices that we've identified for doing this. So first off, let's start with quality control. The data that you're getting may have different quality issues. So there might be poor quality cells, and there might also be some genes that are not actually expressed at all in your data, and that you may want to filter out to reduce its dimensionality. So poor quality cells could be, for example, cells that have a lot of mitochondrial reads expressed, and that essentially means that these cells might have been super stressed, or actually they might have been already kind of partially broken up during the sequencing process, and you may want to remove those cells because they would skew your data in a certain direction that you may not want to look at. In addition, you would also maybe like to filter out cells that have a very low number of counts, because that tells you also something went wrong during the sequencing or during the preparation of the sample. And you may also want to exclude cells that have a very high number of counts, because these could be doublets, or these could just be really abnormal cells that, again, you don't want to have in your data when you're doing subsequent analyses. Yeah, there is a quite convenient Scanpy method that calculates quality control metrics, and this will calculate these entries in ops and in var that you could use to determine your filtering thresholds. So this would calculate the number of counts, the number of cells, and the mitochondrial reads, and yeah, other quality measurements like this. And yeah, I should also mention there is, of course, another lab coming up for this data pre-processing and quality control as well. And this is where you can actually try out all these functions and try and figure out yourself the best thresholds for filtering data. Or you can also see the effects that it might have on the downstream analyses if you don't do this filtering, because you may have, for example, some extra clusters popping up if you don't filter low quality cells beforehand. Yeah, in terms of best practices, we would advise to actually use some very lenient cutoffs, because you may not want to, you don't want to bias your analysis against really small subpopulations. And it could, for example, happen that you have a really small cluster of cells, a cell type that is not very heavily represented in your data that actually has on average a low number of counts. So you don't want to filter all of those cells out because you might actually be interested in them. So what has, in my experience, been a good strategy is to actually start with very lenient cutoffs, do the analysis up to the point where you're annotating your cell clusters, and then figuring out if you might actually have a cell cluster of low quality cells in your data still. Because if you have this, this tells me I need to be more stringent in my cutoffs. So there's this kind of an iterative process that I'm going through usually twice to really clean all of my data. Yeah. Another aspect of quality control is actually doublets that you may have. So I've already mentioned that cells that have a really, really high number of counts might be doublets, but these are not the only cells that might be doublets. So a doublet essentially occurs when you have two cells that result being in one bead or in one drop and are kind of sequenced as a single cell. And essentially, this results in cells they might have a slightly higher number of counts, but due to the, at least if you're using 10x, this is not necessarily the case because you have kind of a fixed number of counts that you can get overall, I think, or something like this. So this is not like a linear relationship. So that you need some more advanced methods to really robustly identify these doublets. And yeah, one way to do this is to essentially simulate doublets from your data that you have. So essentially a doublet will show up like a cell that kind of looks like one cell, but also looks like another cell. So it's kind of a mixture of the two cells. And this is what you're essentially simulating by, for example, using scRNA. And the simulation essentially takes two cells, averages them, and puts them back in your data. And then you can identify likely doublet cells that cells that have many simulated doublets as neighbors in your KNN graph. And this way you can kind of identify those cells that are very likely doublets. And this is essentially what scRNA does. But there is, of course, other doublet detection methods. And yeah, and I'm actually not sure if there's a lot of benchmarks for this, but yeah, there's a lot of different methods. I also use scRNA essentially, but again, there's many different ones. Might also depend on your data, which one is the best here. Yeah, another important point to mention here that doublet detection methods should actually be run on the individual samples that you get. So it shouldn't be run on kind of an aggregated data frame that already contains multiple batches or multiple samples, because essentially doublets can realistically only occur between cells that are in the same sample, because they cannot occur across sample. So if you want to simulate the doublets adequately, you need to consider this in your simulation, because it considers the actual ratio of cells in every cluster to simulate the doublets, and you don't want to mess this up by actually presenting data in a way that it couldn't occur in real life. Right. So okay. So now we have a really nicely filtered data matrix where all the low quality cells, all the doublets are excluded. So to continue with our analysis, we want to look at normalization. So in these count matrices, the issue is that the count depth for the same for really identical cells can differ just because of the variability in the experimental setup. And so you need to be careful if you just compare gene expression between cells based on count data, because any difference might just be due to the sampling effects that you have in your experimental setup. So in order to actually be able to compare gene expression levels across cells or between cells, we need to normalize our data before. And what this normalization does, essentially, it scales the count data to obtain kind of the correct relative gene expression abundances between the cells. And the way, or we have a quite simple method in Scanpy to do this, which is the normalized total method. This essentially estimates the count or the size factor by just looking at the total number of counts in each cell and normalizing by this. So this is a very simple way of doing this. Yeah, and our guidances for best practices here would be to try this relatively simple normalization technique, and then just look at the results on a UMAP and look at the total counts in highly expressed genes in your data set, and look at this whether they actually make sense in a biological meaning. And but there's, of course, also more advanced normalization methods. We tend to use the scRNA normalization, which essentially allows you to estimate size factors for individual cell populations, and then deconvolve these size factors to the individual cells again. And this essentially results in just a much more robust estimation of size factors. It also takes a bit longer to run, but yeah, if you want to have clean data, maybe that's something you should consider. Yeah, there's also, of course, other ways like the SC transform that you could also try and use, especially if you have many rare cell types in your data that are not recovered when using the scRNA normalization. Okay, right. So we have processed and normalized our data, and now let's have a look at what we can do for feature selection. Feature selection means selecting some genes or some combination of genes, or more generally speaking, of features that we want to look at. And there is a couple of reasons of why we actually need to do feature selection. And one is that there is that different features might have a different signal to noise ratio. So some genes might not contain much information at all. And if you use, let's say, 20,000 features, because that's like an average number of genes here that you may have in your experiment, this is just a lot of features, and you may not have so many cells. So you are really running into this curse of dimensionality, where any analysis method, any machine learning or deep learning method that you may want to run in your data, it has a lot more features than actually samples. And you cannot really estimate things from this anymore. I mean, first off, fitting the parameters is very hard when you're this underrepresented. But also, just if you have a lot of features, the data, the distances between your individual data points tends to become very, very similar. And this is what is known as this curse of dimensionality. And one way to counteract this is really to reduce the number of features that you're looking at. And this way you can actually get some differences or some different distances between your cells to, for example, calculate good KNN graphs to do any sorts of analysis afterwards. Another point for this, of course, is to just computational and storage efficiency, because it is a difference whether you're looking at 20,000 features or just at 50. So what can we do to select features? Well, one approach here that one usually takes is to select those genes that have a higher variance than expected from the standard mean variance relationship. So these would be the highly variable genes. And in order to get these highly variable genes, what we're looking at is the relationship of the dispersion to the mean. So the dispersion here is simply just the variance divided by the mean. So this is what is known as the dispersion. And essentially, you can see this in the upper right corner here, where we're plotting the dispersion against the mean expression of each gene. So every dot here is a gene, and in red are the highly variable genes. So the red color means these are the highly variable genes. So you can see these ones have a higher dispersion than expected from the mean expression. So they're kind of not on any diagonal in this plot here. And the way you would calculate them with, for example, Scanpy is with this preprocessing function, highly variable genes. And what this function does is it bins the dispersions by the means. So it computes this histogram of means and dispersions and normalizes them. So just doing a z-score, and then just takes the top genes based on these normalized dispersions. So these are the ones that are, well, that have a larger dispersion than expected. And yeah, what we usually do is, depending on the dataset complexity, you would maybe use between 1,000 or 5,000 highly variable genes. You can also use less. It really depends on what kind of data you're looking at. This is now spoken only for the RNA, single cell RNA data. Yeah, another important point to note here is that if you use this highly variable genes calculation based on the dispersions, you need to make sure that you have not yet normalized your expression values to, for example, 0 mean and univariate. So you have not yet scaled your expression variables, or that you haven't yet used the S3 transform, because this essentially changes the distribution of your gene expression. So essentially, it's important to do this highly variable genes calculation before you do any of these more advanced processing steps. Right. Okay, so finally, we have cleaned up our data enough so that we can actually have a look at it. And yeah, there's many different ways to visualize data. There is PCA, principal component analysis, which is a linear way of reducing the amount of information. There is t-SNE and UMAP, and there's also diffusion maps or force atlases or any sorts of things that you could use. And yeah, what we usually do is we use UMAP just for exploratory visualization, for looking at where clusters are and how they might relate to each other. The PCA for a really good summary of your data, where you can be sure that the data is not skewed in any way, because it's a linear way of reducing the dimensionality of your data. And diffusion maps are really helpful if you're looking at trajectories in your data, because this is what they are meant to emphasize in your data. With all of these techniques, it's important to mention that you shouldn't over-interpret anything that you see in any of the nonlinear dimensionality reduction maps. So for example, you should not just look at the UMAP and kind of start to measure distances between clusters on the UMAP and use this as a proof that your clusters are different or the same or things like this, because it's a nonlinear way of reducing your data. So it inherently needs to overemphasize or underrepresent certain types of distances in your data. That's just the way it is. So any types of quantitative results should always be based on, well, on the actual data and not just on the UMAP. But granted this, it's a very useful tool to actually look at your data and get a feeling for how, for example, individual cell clusters relate.
Hanna, sorry.
Yes.
Can I ask you a question about the, you previously shown the quality parameters, right? So you've used the total count, but also mitochondrial content.
Yes.
So what do you generally use as, are these two that you generally use, or what other factors do you look at? And do you use regression when you have identified your variable features?
Okay, so so the first question for the quality metrics that we calculate, so the number of counts and the percent mitochondrial reads is mostly what we use. I also look at the number of cells per that every gene is expressed in, and I filter those, although it does not seem to show that many benefits. Maybe I'm a bit old school for this, yeah. But yes, these are mostly the ones that I use. I also look at the cell cycles. If I'm interested in, I mean, that depends on what kind of subsequent analysis I want to do at some cell cycling scores, because maybe I'm not interested in the actual cycling cells, because that might confuse my results.
Exactly, yeah, that depends exactly as well on the in and out type of story. So
yes.
You don't generally use the ribosomal content. It's not something you look at.
No, this is actually also something you would look at. Yes, yes. So okay, yeah. Yes. Sorry.
And that would include regression as well, or no? That would just be the quality first, or the filtering, or would you also regress for those factors?
I mean, I would at a first step just do the filtering, but again, I mean, I think it depends on what kind of questions you have and what you expect from your data to, yeah, what you expect to be there in your data. It's like very basic steps. I would do filtering thresholds. Yeah. But yeah.
No, I get your point, because exactly that's what I'm hoping to get some type of consistency with it, because it's so flexible. I'm just looking to find out indeed what type of thought patterns you use. But yeah, I understand your point. Yes, great. Thank you. Yeah.
Awesome. Great. Yeah, thanks. Let me know if there's any more questions, please. Okay. Yeah. So we were at visualization. So I just wanted to show you a few other ways of how you can visualize the data. So of course we can do like UMaps or other types of 2D projection methods that I've shown you on the previous slide. But we may also want to look at yeah, the distribution of gene counts in individual clusters, for example. So we could look at violin plots, or more generally, maybe if you want to compress this information down, you could look at dot plots where you just visualize, let's say, the mean expression. These dot plots might also be used if you have some differentially expressed genes and you just want to see which ones are significant across individual clusters. You can do this with the dot plot method. There's also a way to show kind of small violins. This is similar to the dot plot style. This would be the stacked violin. We also have matrix plots. More in general, you can put in all kinds of information in this. And what I also find quite useful is to look at the heat map plots. This essentially does not show a summary, so the mean of all the cells in this particular cluster that the dot plot or the stacked violin would show, but it actually shows the expression of every cell if you group by clusters, for example. So this is what you see in these really really thin bars here. So you can see how the how the gene expression varies even within individual clusters. Right. Good. So finally, this is kind of the last big point that I wanted to talk about is clustering, and also cluster annotations, because so far I've kind of focused on cleaning up our data and processing it in a way that we can handle it. And now comes the step of where we actually start to look at what do we have in our data, is what kind of interesting features do we find there. And one step, of course, that we always or usually do is clustering. So what we do is we would recommend using the Leiden community detection method. This is based, so this essentially detects communities in the KNN graph. And the KNN graph is usually computed based on, let's say, the highly variable genes, and usually using a PCA of the highly variable genes to deal a bit with this problem of the curse of dimensionality that I mentioned before. Yeah, and essentially we can do the clustering at different resolutions, and you could subcluster to get kind of a better handle on smaller subpopulations that you're really interested in figuring out. Unfortunately, there is no automated one-fits-all sizes solution for this. But also in my experience, clustering is something that I usually need to do a couple of times to get a feel for the resolution that fits for my data. Because also the resolution that you give this Leiden algorithm depends on the number of samples, so the number of cells that you have. So if you use less cells, you may need to use different resolution. That's just the way this clustering algorithm works. Right, but after we've identified kind of interesting potential groups of cells, we want to annotate them with some biological meaningful names. And again, this is something that is usually an iterative process where you start with maybe a coarse naming of your clusters and more refine this the more you learn about your data set and what kind of variability you have there. Manual annotation is usually based on some marker genes that you have from literature or that you know are relevant for previous experiments, and then you can look at a dot plot like this shown on the slide to figure out which of the relevant genes are expressed in what cell, in which clusters, to kind of get a feeling for which clusters may represent which cell types. Like I mentioned before, this is quite a time-consuming process. So if you have the opportunity of using a reference atlas to speed this process up, this is a really great opportunity to leverage kind of all of the information that the community has gathered before, and you could use that. So essentially, the most basic way of using a reference atlas would be to, for example, just integrate both your data set with the reference data set and just look at the neighbors of, so look at the nearest neighbors of each of your cells, note down which clusters they are in, and just transfer the annotations from your references in this way. But of course, nowadays there's many more advanced methods to do this type of semi-automatic annotations, and they're really worth checking out if there is good references for your data set. Other than that, I'm afraid there is still like a manual step that you will just have to do if you want to give your cluster some biological meaning. Right, so these were essentially all of the topics that I wanted to cover. I've just noted down 2 more that I think are quite important, but just haven't quite fitted in this presentation. And one is to actually correct for unwanted variation in your data. So this might be if you have different biological samples, or even just different technical replicates of your data. They're just due to the fact that they were sequenced at different times. Maybe the temperature in the lab was slightly different. Maybe the technician did something a few minutes later than it's usually done. Like all of these factors may result in variation in batch effects in your data that essentially might hinder your ability to analyze all of these data at the same time. And for this, what we usually use is some batch integration, some integration methods like Scanorama, SCVI, TRVAE, and there's many more. I mean, I'm just mentioning a couple of the Python tools that we usually use, but there's a lot more that one can use to essentially reduce the variation of batch effects in your data. yeah, I just want to point you to a recent benchmarking paper for some of these algorithms that you can have a look at to figure out which one would be adequate to use in your in your data. And just make you aware of this fact that almost certainly if you have data that comes from different samples, and you should have, because it's always good to have technical replicates of your data, you may need to integrate it to use some batch correction method before using it. And I think in the lab there's also an example of how to use, I think it's Scanorama, some sort of batch integration to on individual samples. So this will be really really good for you to have a look at as well. Yeah. Finally, just a point about differential expression here. I already mentioned that differential expression is something we could, for example, use to figure out which genes are, well, differentially expressed, like the name says, in individual clusters to, for example, facilitate cluster annotation. There is quite simple tests like a t-test or a Wilcoxon rank sum test that are implemented in Scanpy that you can directly use. This is the rank genes groups function. But there's also much more powerful tests that you could use for specifically developed for single cell data. However, it could be that differential testing on individual single cell level is overly confident. So it's actually, it might be recommended to use pseudo bulking to get more statistical power and more reliable results from these differential expression methods. So pseudo bulking means you may, for example, average the gene expression per cell cluster, and then you can use kind of traditional methods developed for just omics data, like the DSEQ2 or the HR or the, or all of these types of algorithms. I'm also here showing you a paper where this phenomenon is described. I just want to mention this because sometimes in the analysis I get the result that there is, I don't know, 2,000 differentially expressed genes. And when I actually look at them, when I visualize them on the UMaps, I can see that there is very little variation. And there might just be kind of some outliers in my clusters that are driving this result when I'm looking at the individual cell level to do these tests. So just again, take all of these results with a grain of salt. And it's always good to visualize the data afterwards and to see if your results actually make sense, if you can actually see them by eye in the data as well. Right, so this is everything I have to share for now. If you have any questions, I'd be happy to take them.